Great. Other questions? Okay. Then we were talking about protein folding at the end of last lecture. And I wanted to point out there's a type of protein that actually helps other proteins fold, and they're called chaperone proteins. The one at the right here, the left here is called GROW-EL. I think it's like that. GROW. But I think it's an E. coli protein, if I remember correctly. But it's kind of like a big garbage can in terms of shape. If you look at this thing, what it has is it's got a big space in here, and it's kind of got a cap on top, and place here where you might thread your protein in. So what happens is that the ribosome gets associated with this, and it's kind of synthesizing and threading the protein into here, and then the protein can fold in this space where it's protected from other proteins of the same type or other proteins that might have hydrophobic patches on it. So one of the problems with protein folding, one of the problems you see in a lot of these various brain diseases where you're getting, we talk about plaques and things like that, is that you have proteins precipitating out. What they do is you have a sticky hydrophobic piece on one protein, sticks to another sticky hydrophobic piece on another protein, and now they can't fold. And then another sticky piece comes in, and another sticky piece comes in, and eventually you end up with something that's kind of too big to be held into solution, and it crashes out. You get enough of that happening in a cell, and the cell dies. If it's happening in your brain, you can start losing brain cells, which is not a good thing. Okay, so that's generally what happens. This is actually like an E. coli chaperone. But you have these sorts of things. If I have a protein that's particularly hydrophobic and there's danger of it sticking to other hydrophobic proteins, you can synthesize it into this space, and that it can fold in this space and be protected from other proteins. And then this yellow cap, once it's done, this yellow cap can open and the protein can come out. Does that make sense? How that works? Okay. All right. If you're having trouble remembering, chaperone proteins are here, right? What they're trying to do is prevent inappropriate aggregation, right, between proteins. Same thing that chaperones do on dates. Okay. All right. Anything else about protein folding? Just kind of finishing that up. We're going to then talk about how you can purify proteins. Okay, and we're going to start off talking about chromatography is often used, and tomorrow we're going to use chromatography in lab, and we'll talk about the specific method we're going to use tomorrow here. So chromatography is often used. And if you remember from our discussion in the lab with the LEAF lab where we talked about TLC, we're going to have two phases. You have your stationary phase, which is not moving. Okay, often in terms of column chromatography, that stationary phases, we call them beads. They may be, we can't see them. They may be nice and round. They may not be so nice and round, but we call them beads. All right, and then we have the mobile phase. We have the stationary phase, and then we have a mobile phase. The mobile phase is the phase that's moving down the column in the case of column chromatography. I'll just say moves through the stationary phase. All right, and proteins are going to be separated based on their relative affinity to the stationary phase versus the mobile phase. And I'm going to say, I want to make a point here from something I was reading in your some of the leaf labs. It's their relative affinity. When I say relative to affinity, relative to each other. All right, so what's going on with that? Why do I care about relative to each other? The relative to each other means that, it could be all of your proteins on this column have a stronger affinity for the stationary phase relative to the mobile phase. All right, and so they all have a stronger affinity for the stationary phase, but protein A has more affinity for the stationary phase than protein B, if that makes sense. Okay. So what I was seeing on some people's lab reports in describing this, they talked about, well, things that are nonpolar are in the mobile phase and have a higher affinity for the mobile phase, and things that are polar have a higher affinity for the stationary phase, right? And it's not that clear cut. Something that have a higher affinity for the mobile phase is probably really running with your solvent run. Okay, it's probably running super fast. Unless those two affinities are really close. So remember that it's relative to each other. Okay, so there are various types of column chromatography. One type, okay, we're going to start with ion exchange chromatography here. One type is ion exchange chromatography. All right, we have two types of ion exchange chromatography: cation and anion exchange chromatography. All right, so we have these two types here. In cation exchange chromatography, the cations are exchanging off the stationary phase. The cations have affinity for the stationary phase. What do you think the charge is on the stationary phase? It's negative, right? It's a little confusing, right? It's called cation exchange chromatography, but the stationary phase is negative. We'll just say the stationary phase is negative, and the proteins, the cations exchange off it, we could say, and the proteins exchange off it, we'll say. So what's going to have the highest affinity for a cation exchange column? Positively charged proteins. So the more positive the protein, the more affinity it has for the stationary phase. And the faster or slower it will move down the column. The slower it will move down the column. Right. Okay, because it has a higher affinity for that stationary phase, it spends more time on the stationary phase and it moves slower. Remember, this is called ion exchange, but and like any of these chromatographies, or like most of these chromatographies, actually, I should say, things are exchanging on and off. So it's on the stationary phase, it comes off, and it moves in the mobile phase a little bit, and then it's back on. And it's how long it's on that determines how fast it moves down that column. Anti-exchange chromatography is the opposite. The stationary phase will be what? Positive. And the proteins exchange off it again. So the more negative the protein, the more affinity it has, and the slower it moves. And so we have an example of this down here, a cartoon for it. If we look at our column here, all right, we'll go and look at our key here for what's what. So this is a column. Our beads in the column are negatively charged. And so something that is, the red is large and positive, or large positive, I should say, large positive charge. And blue, dark blue is most negative. And so what you see is you'll pour these things into the column, and our very positively charged things will stay at the top of the column and they won't move very much. It is possible with these things, just like you saw with your TLC, the red dye stayed at the bottom, right? Because it had a very high affinity for the stationary phase. It's possible to have something that has so much affinity for the stationary phase that it would just stay up here. And then our very negative things, they're running down that column as fast as possible. They're spending very little time, if any time, on the beads. Okay, they run through very quickly. Less negative, spends more time. Now we're getting to a little bit positive, and then very positive. Does that make sense? And so that's how you would see that. Is you would be collecting aliquots or samples at the bottom of your column, and then you could detect whether there was protein in them. How might we detect whether there was protein in those samples?
Using like an indicator, like I think there's a one. It's using the Bradford assay. It's a dye that changes color when.
Okay, so we could use the Bradford assay. Bradford assay is a dye that binds to proteins and changes color when there are proteins around. Now, if this is my precious protein sample I want to study later, I'm not going to want to add Bradford to that. I could take a sample of it and run a Bradford on it, but I wouldn't want to add Bradford to it because it's destructive. It would wreck my protein or wreck functionality. What else could I do?
UV vis.
UV vis, right. So in lab, we've detected proteins already. Remember you did your BSA the first lab? We detected the BSA protein at 280 nanometers. Okay, so we could do that. Questions on ion exchange chromatography? Okay, so our next one is going to be size exclusion chromatography. And this is something we could have done instead of dialysis. And in fact, next semester in lab, we will do it instead of dialysis. So in size exclusion chromatography, you have beads that have a bunch of little holes and channels in them. All right, so they're holy beads, they have channels, and the proteins can either fit into those holes or not fit in those holes. Okay, a protein that fits into the hole is going to kind of go down. It's going to go into a bead. It's going to run around for a while. It's going to come out of the bead. It'll go down some more. It'll run into another bead. It'll run around for a while. So something go into the holes is going to move slower or faster down the column. Slower, right? Because it's going to spend time hanging out in those beads. Okay, so this actually is going to separate things in a counterintuitive manner, right? Larger is going to move faster. You're used to everything else, right? You're used to gels and things like that. Everything else smaller moves faster. Okay, so let's just write down how this works then. So the beads in this column have lots of channels. Okay, small proteins can fit in those channels while large proteins cannot. I'm going to say other molecules, because you might just be looking to purify your protein away from something you were using, right? So our dialysis, what are we trying to purify with our dialysis in lab this week? LDH, lactate dehydrogenase, is our protein we want, right? So we're not trying to purify that away. What are we trying to get rid of? You guys remember? You did an ammonium sulfate precipitation, or try to get rid of that ammonium sulfate, or get rid of enough of it that it doesn't wreck our next column, the column we're going to run tomorrow. So small proteins and other molecules. So could it be that these other molecules could be your ammonium sulfate, can enter the channels, and this causes them to move more slowly down the column. Than large molecules that just run around the outside of the beads. And so down here we have an example of that. Okay, so what you've got here is they're showing you these porous beads, although they're not really showing the pores and the molecules going into the pores such that they match in terms of size. But imagine you have these porous beads here. They're actually showing these proteins in terms of their size. The big proteins are just going to run around the beads. They're just going to run past the beads here. The medium ones might be able to enter the channels a little bit. And then the small ones can kind of enter the channels. And so they're going to have a much longer path going through this column because they're going to be going down and going sideways and running around before they get to the bottom. Okay, questions? And once again, you'd get to these things down at the bottom. If the protein didn't have any kind of inherent color, which your LDH, your lactate dehydrogenase will not, you're going to need to do some sort of technique to detect whether your protein's in these tubes or not. And we are going to use UV vis. Questions? Okay. All right, finally, this is the type of chromatography we're going to use tomorrow. This is called affinity chromatography. And this is great at getting a really quick and efficient purification. Okay. For other things, we were taking a physical property of protein that many proteins have, right? Lots of proteins are big, lots of proteins are small, lots of proteins are positively charged or negatively charged. But here, what we can do with affinity chromatography is we can put on our stationary phase a ligand. Okay, so ligand is something that something else binds to. In biochemistry, the ligand is some molecule that your protein's binding to. And so in affinity chromatography, you put a ligand on your column that your protein binds to and either no other proteins bind to or very few proteins bind to. So a ligand that is bound by the protein of interest and not many other proteins is attached to the beads of the column. Okay? You then pour your protein solution down the column and the protein will bind. Okay? You wash the column. So we have this column, we poured our protein down, it's stuck. So that's what's showing here. I've got a mixture of proteins. I pour these proteins down the column, and my red moons are going to bind to my, whatever that is. I've got my red crescent moons, and I have my little circles. My red moons bind to all the circles. Everything else just goes out down the bottom of the column. What they don't show here is I wash the column with something else a couple times. Just make sure I get everything else off of it. And then the way I'm going to elute my protein is I'm going to put in something it also binds to very specifically in the same spot. So for this cartoon, they took that ligand, they bound to the column, they also had a solution of the ligand, the exact same thing. When we do it, we're not going to have exactly that. We're going to have a column that has a nucleotide on it. That nucleotide is going to be bound tightly by your lactate dehydrogenase. And then what we're going to do is when we want to elute your protein, we're going to bind your protein, we're going to wash it a few times, and then to elute your protein, we're going to add a solution with another nucleotide. But it doesn't have to be the same thing on the column and that you're eluting with. But both things have to be things that bind to your protein of interest very specifically and tightly and in the same spot. Okay? Why is in the same spot important?
Otherwise it can stick to both.
Right. Otherwise, your protein could stick to both. If we had a situation here, so let's imagine I've got a bead here, right? And it's got my ligand bound there, and my protein of interest binds to it like that. And if I add a second ligand, then it binds super tightly, but that ligand binds over here, it's not coming off the column, right? The way I get it to come off the column is I give it something else to bind to in solution. And often I'll throw that in at a pretty high concentration relative to the other thing. But really, all I need to do is, if it's bound really tightly in a column, all I need to do is get it to, occasionally go out and bind to something else, and it should come off the column pretty well. Questions? Let me kind of finish up this description then. So what did I say up here? A ligand that is bound to the protein of interest and not many other proteins attached to the beads of the column. The protein solution is poured down the column and the protein binds. The column's washed to wash off all non-specific proteins. Then another ligand that the protein of interest binds tightly is added in the mobile phase. The protein binds the ligand in the mobile phase and comes off the column. And we're going to say elutes from the column, okay? And And let's go back and just specify the column is another ligand that the protein binds tightly at the same protein site as the ligand on the column. Is added in the mobile phase. Does that make sense how that works? Okay. Does the second ligand need to be really specific for your protein? And does it need to not bind a whole bunch of other different types of proteins?
No.
It doesn't matter, right? Because all we have on the column at this point is our protein of interest. So if this thing bound to every protein in the world, but also bound to this protein and at the correct place, it would still work well. We've already washed everything else off the column. I don't know how often that happens, but in this case, if you didn't care about the function of your protein, you could like just heat the heck out of it, right? And what's going on here is, so they don't show a really good picture of it, but this is more like your red pigment on your red leaves. Remember, it didn't move, right? When you do this, what's going to happen tomorrow is you're going to pour your proteins down. Your red protein is going to stick to your column, possibly all the way down like this. We won't know because there's not a color to your protein. And that it is not moving down that column until you add a ligand in the solution. So it's not slowly moving, it's just not moving until you add a good ligand in your mobile phase. Okay. And tomorrow we will use UV. Like I think I already said that, but questions? All right. Let's talk about some gel techniques then. So one gel technique that gets used for proteins a lot is polyacrylamide gel electrophoresis. Okay, and this can be used to separate proteins by size. So how does this work? So we have a gel like we did. We have a polymer, and this polyacrylamide is just the polymer that's used. For our DNA gels, we used agarose as our polymer. All right. The difference with polyacrylamide is you actually polymerize. So the agarose gels were like Jello. You heated it up, you cooled it down. It was like Jello. It spread out, it trapped water, and it made a gel. For the polyacrylamide, you're actually polymerizing the acrylamide. Do you remember polymers from polymerization from organic? You're doing a polymerization reaction to make your gel. But it's the same thing. You still have a gel mesh. Okay, so a gel mesh slows the protein down as it moves through the gel. All right. Now once again, we've got run towards red like we did before. Why did your DNA run towards red? Yeah, because it's negatively charged. Are all proteins negatively charged? No. So we need a way to fix that. Okay? Also, are all proteins the same shape? No. We also need a way to fix that. All right? So what we have is a problem that not all our proteins, if we have a positively charged protein, it's going to run out of your well this direction. Right? That's not good. You don't want to lose your protein. You want to see it. And so what we do is we add something called sodium dodecyl sulfate to our gel. So let's imagine we have some proteins and they look like these curly Qs. What we're going to add is a molecule called sodium dodecyl sulfate. Okay, so it's sodium. Actually, I've got the wrong. You know, I can never remember whether I attach with just a carbon. Somebody can look that up for me. I think it's this. But it's dodecyl, so it's 12. 2, 4, 6, 8, 10, 12. Okay. And so what we have here is we have a molecule which has, we've got a negative charge, right? This is polar and negatively charged. And then this part right here is nonpolar. So about half of the amino acids in a protein are nonpolar. And what happens is overall about half of those amino acids are going to be bound by SDS. So you'll hear this called SDS. And so if we come down here and draw a little cartoon about what's going on, we've got, so we're going to add SDS, and I'm going to make my SDSs like this. Actually, I used green up there, didn't I? Let's do some little green bands. Okay, I'm going to add my little SDSs like that, and they're going to go in here, they're going to bind to the protein, and as they do that, they're going to cause it to unwind. Okay, why would they cause it to unwind? It's a detergent. How more specific?
Oh, it messes with the covalent interaction or non-covalent interaction with the protein.
It's going to mess with the non-covalent interaction. So we have these van der Waals forces, right, where it's hydrophobic. I want more. That's all I got. What are these? These are this.
Is it going to break the hydrogen bonds?
Probably break hydrogen bonds too. I want more than that.
Increasing actually different parts of the proteins are attracted.
It's negatively charged. And I'm putting a whole bunch of them next to each other, right? If I have one every other amino acid, that's a lot of negative charge in there. So the protein's going to unfold and stretch out to get those negative charges as far from each other as possible. All right, so what's going to happen, what this is going to look like, let me keep this all on screen, is our proteins are going to unfold, and they're going to unfold because the SDS is going to get in there, and it's going to bind every other amino acid. And I'm going to have these, I'm drawing them as red, but these are negatively charged groups. And they're going to push against each other, and that's what's going to help keep this thing unfolded. We've got all those things going on, but the negative charges all next to each other are going to do a great job of keeping this unfolded. Okay? And they're going to be attracted to this positive charge on this gel that's kind of half off the screen here. All right? So what now we have is we have all of our proteins are negatively charged. All of our proteins have one negative charge per every other amino acid pretty much, because proteins don't have such a huge charge that it matters much compared to every other one having a negative charge on it, right? It may be slightly different, but it doesn't matter that much. So now I've got every other amino acid has a negative charge, so these things now move based on their molecular weight. And they're all the same shape, they're linear. Okay? Does that make sense? So it's sodium dodecyl sulfate. Okay, is added, and that's SDS if you ever hear about it. I wonder if I actually spelled that wrong. No. Okay. Sodium dodecyl sulfate is added to the protein. It causes it to unfold and gives it a uniform negative charge. All proteins unfolded are also linear. Okay, and so everything moves on the gel based on its molecular weight. Questions? All right, and so if you were to look at one of these gels, this is what it would look like. So this is a cartoon of what one of these gels would look like. That's a cartoon where you were maybe looking at something very pure. So you do these the same way we did our agarose gels. We had a molecular weight marker where we knew how big things were. And so you might have some unknown protein here. Obviously, this is super pure. If you saw this on a gel, what you generally say in the literature is you say your protein's by gel electrophoresis is 95% pure. You can't see anything else on it. That's about what you can claim. Okay. But so this protein here would be about how large? Sophia?
38.
38, sure. 38 kilodaltons. Not 38, yeah, 38 kilodaltons. Okay, is what we would call them. And so over here, we don't actually have a molecular weight marker, but this is an example of a gel where they're clearly doing some sort of purification. This might be a cell extract right here, and then as they move their way over, maybe they purified out some parts of it, done some sort of chromatography on it. Here would be something different. Here would be something different. Okay, but you can see that they might have pulled this stuff out. Might be that band. It's a little higher, but as it purifies out, it runs a little bit differently sometimes. Questions on how that works? All right, so we have a gel technique now where we can separate the proteins based on their molecular weight, but you can imagine when you look like in here, there might be things on top of each other, right? You can almost see things on top of each other in some of these places as well. So it might be difficult to use this gel to actually kind of pick off specific proteins. So one way you could help that is you can add another dimension to your gel. This is a one-dimensional gel, right? Well, we could do a two-dimensional gel. And what our second dimension could be is we could take advantage of PI for proteins, okay? So you can use isoelectric focusing to separate proteins by their PIs, right? We talked about that, to add another dimension. All right, and so this, like I said, you would separate them by their PI values. That's what we see here. We might add some protein here, and then we would turn on the electric field, and then these proteins will move throughout this pH gradient until they find their PI, which is where they have a net charge of 0, and then they stop moving, because if they have a net charge of 0, they're not attracted to the positive electrode or the negative electrode. Could I have loaded them down here? Yes, because they would have moved. I could have loaded them down here. They would have protonated based on their pH. In fact, in this case, they would have mostly been positive, okay, because the pH down here is going to be, well, I didn't even need this, right? Here. Which pH is at the bottom? Okay, the low pH though, right? I know the low pH is going to be at the bottom because that's where I'm going to need my positive charges. And to get positive charges, I need to have lots of protons around so they stick to things. So I could load down here, everything would be positive. It would move until it became neutral, and then it would stop. Could load anywhere on that gel. Okay, so what you would then do, so we can separate proteins by their pI values, this allows us to add a second dimension or do a 2D gel. And so here's a picture of what that you would do with this. Whoops. All right, so you do your isoelectric focusing gel, you'd lay it on top of your other gel, and then you'd run it down, and then you'd have proteins separated in this dimension by pi and in this dimension by molecular weight, so by size. So smaller in this way, and then here it says decreasing pi is the way they laid their gel on top. And this is what one of these things would look like. And before I started teaching, or I guess in between when I started teaching, I taught a little bit, and then I went to industry, and then I taught again. I worked with a company where we would do these gels, so we'd separate things out in 2 dimensions, and then you'd want to look at what proteins were. So you were looking at perhaps a 2D gel of a normal human cell, or a healthy human cell, I should say, versus a cancer human cell. And then you'd go in there and you'd say, well, that one is on one of my gels and it's not on the other. So that's interesting. It's on the cancer one and not on the healthy one. So let's punch that out. You would punch that out and then you could mass spec and figure out what it was. Okay. So a lot of cool stuff you can do with these sorts of things. Questions on how those work? Okay, questions on protein purification. There are other ways you can purify proteins, but those are some of the really major ones used. All right, then we're going to go on, and we're going to start talking about myoglobin and hemoglobin. We'll probably just introduce myoglobin today. And hemoglobin tends to be, so we're talking about myoglobin, we're working our way up to hemoglobin. Hemoglobin was like one of the first proteins that was really heavily studied for how it works. So it's in all the textbooks. It's the one we teach. It's also really interesting. It does some cool stuff. This is we'll see how much of it I get to on Wednesday, but then this is my favorite set of lectures because hemoglobin is just it's an amazing example of, I mean, it's natural selection doing it, making a molecule, natural selection resulting in a molecule that just responds to so many things in its environment. Okay, but to start that off, what's this? That's a cast iron skillet. What's going on with this cast iron skillet? I love my cast iron skillet. What was that? It's rusting a little bit. Right, okay. So what's the black stuff? Anybody know? On your iron skillet? Iron. Iron what? It's not, iron metal is shiny. Iron oxide. But isn't that rust? So the black, the black on this is iron oxide. Like that, okay? And iron oxide like that is a black paint, okay? They actually use that black paint. Anybody know what the Hindenburg is? What was the Hindenburg?
Was it a blimp?
It was a blimp, right? What happened to it?
A lot of really bad things.
Well, maybe just one big bad
My
thing.
favorite piece of trivia about the Hindenburg is the fact that not all of the people on board died, which was surprising.
That not a lot of people aboard died?
Yeah, it was like 30 fatalities.
Out of?
I forget.
Okay, I don't know how many people you could put on a flip, but it could be not a lot of people died. It was just spectacular in how it burned. But people talk about it. So in World War II or before World War II, or at least before we got into the war, we wouldn't sell helium to Germany because we didn't like what they were doing. We had helium reserves and they didn't have helium reserves, um, and so they filled the Hindenburg with hydrogen gas. All right. So as you might know, hydrogen gas, when you expose it to oxygen, you light a spark, it goes up. It will make an explosion, right? So there are these spectacular pictures of the Hindenburg burning. Um, and it turns out that, you think about, well, the hydrogen blew up and that caused it to burn, but that really wasn't the biggest problem. The problem was that it was painted in this black iron oxide. And when you get that hot, it turns into iron rust fast. And it's a very exothermic reaction. It's an oxidation reaction. So it's at least highly speculated that really the problem wasn't so much that we had hydrogen go off, but that it burned so intensely because of the paint that was on it. Okay, and so what will happen is if you take this iron oxide and you put it into air, just like your cast iron pan, it's going to turn into this iron oxide. Okay. Now it can't be completely that, or I don't know. And the Hindenburg was thin, right, because it was a dirigible. When I think about it, your cast iron pan doesn't start on fire around it, right? That doesn't go all the way around it. So maybe that hypothesis isn't so great, but it is an exothermic reaction, and it is a spontaneous reaction, and this is why your cast iron pan will do this if you're not careful with it. Okay, so what's the formal charge, or what's the oxidation state of iron in this top one? +2, right? So this is going to be iron 2. Whoops, 2, not 3. What's the one on the bottom? +3. So these are our two common oxidation states for iron, possibly the only oxidation states for iron. Ask Dr. Hughes. Okay, these are the only ones anybody ever taught me. So we have these two iron oxidation states, and spontaneously we go from here to here, which tells us what about this oxidation state? It's favored, it's more stable, right? Okay. So what do I do to my cast iron pan to protect it from going from oxidation 2 to 3? Anybody know? You season it. What's the seasoning?
Oil.
It's a layer of polymerized oil, actually. Okay? So you coat it in oil, and it's polymerized, and what is that? How does that help? It doesn't let oxygen get to it. Okay? So we can't go from Fe2 to Fe3 without adding more oxygen. All right? Why am I telling you about this? Because the same thing happens in hemoglobin and myoglobin. Okay? For hemoglobin and myoglobin to work, what's going to happen, or what hemoglobin and myoglobin do, is they use iron to reversibly bind oxygen. So hemoglobin and myoglobin use iron 2 to reversibly bind oxygen. Okay, and they need to use iron 2 because none of those 20 amino acids that you've memorized, right? None of those 20 amino acids actually reversibly bind oxygen. So they need something else, okay? And then when we think about these proteins, but we don't just have iron 2 floating around in our blood, we've got this protein. And actually we have a porphyrin group holding the iron, which is held by a protein. And all of that is designed to help keep iron in that +2 state. Okay? Yes?
Can you explain what you mean by reversibly bind?
Reversibly bind to oxygen? Yeah, so when I talk about reversibly binding oxygen, hemoglobin and myoglobin they can bind to oxygen, and they also have to let go of oxygen, right? So let's actually talk about that. So hemoglobin is an oxygen transport protein. All right, so it transports. It's like the UPS truck. The UPS truck goes and picks your stuff up, right? And it brings it to you. And if it just drove by and didn't drop off your stuff, would you be very happy? No. Would that be a good delivery vehicle? No. Same sort of thing for hemoglobin. It picks up oxygen in your lungs and it has to drop off oxygen in your tissues, or else it's not doing its job. So for hemoglobin, it picks up oxygen in your lungs and drops it off in your tissues.
Is that going to be too much oxygen? Is that going to convert it to S3 after? Because it's picking up oxygen.
Well, that's a good point. So why, if it's picking up a bunch of oxygen, and it actually in your lungs, assuming your body's operating correctly, if you've ever had one of those little oxygen things they stick on your fingers, right? They're checking. They're looking for it to be above 9%. Usually it's above higher than that even. So for probably everybody in the room, your lungs are oxygenating your blood up to 95% of capacity or better right now. Okay. So it's going to pick up, it picks up that oxygen, but it has to stay in the F2 or the iron 2 state. And so we're not going to have time to talk about it now, but we're going to spend a bunch of time talking about it on Wednesday about what parts of the protein keep that oxygen from oxidizing the iron to the +3 state. Okay. It's really important that it stays in the +2 state. So why don't we leave it at that? But we'll talk a lot about that on Wednesday. If anybody wants to see me, come let me know. I will post the answer key to the quiz, but I'm